Hi guys, we're back for another video. Today we're gonna solve February March 2022 Biology 9700 variant 1-2. Okay, this is the latest paper. I'm gonna attach the threshold and question paper with mark scheme below in the description. So A was at 24 actually, quite low. B was at 20, that means half the marks. Well, C was at 17, D was at 14, and E was at 12. Okay, so in this paper, since there's a difference of 16 from full marks, the A star was probably at 32. So if you get 32 here, it will be hidden, but you'll probably get an A star at around 32. Okay, so let's begin. And if you like the content, remember to subscribe to the channel. Alright, let's begin. Starting with question number 1. What do we have? Which cell organelles are clearly visible when viewed with a light microscope at high power? Ribosomes cannot be seen. Remember that, okay? Ribosomes cannot be seen with a light microscope. So we're excluding A and B. We can see chloroplasts though. Yeah, so C and D are possible. Now, what about centrioles and endoplasmic reticulum? We can't actually see them, okay? So the answer is D. We cannot see these three with the help of a light microscope. Microvilla and root hairs are characteristic structures of some cell types. Which row identifies some features of a microvillus and root hair? So the root hair is surrounded by a cell wall. Yes, it's a plant cell basically. And more than one present on a cell, yeah, that's microvillus. But there can only be one root hair on one cell, okay? The photomicrograph of a plant cell is shown. The cell is 25 micrometer in width from X to Y. So basically I can't do this, right? Because I don't have a scale with me. But if you do print it out, I'll tell you what you're gonna get. We know that magnification is equal to image length by actual length. Okay. So it was gonna be approximately 5 centimeters. This is around 5 centimeters. If you do, if you do find out using your scale. Now we know that m is equal to I by a. So the image length is 5 into 10 to the power minus 2. Actual is 25 into 10 to the power minus 6. Now if you do solve this, you're gonna get 2000 or 2 into 10 to the power 3. Okay, answer is C. Moving on to 4, which eukaryotic cell structures all contain nucleic acids? Okay, so basically nuclei do contain nucleic acids. Nuclei contains nucleic acids. Golgi body does not. Chloroplast and mitochondria do contain nucleic acids because of the endosymbiont theory. They were mainly bacteria. They have 70s ribosomes, right? That's the logic behind this. But centrioles don't. Okay, centrioles don't. So that's why D is the answer. The nucleus is situated in the cytoplasm, right? So technically the cytoplasm does contain nucleic acids, right? Plus there's tRNA, right? That's a nucleic acid. Which statements about viruses are correct? They contain DNA or RNA. This is fine. They use host cells to synthesize virus proteins. This is true. They can have a protective coat of peptidoglycan. Wrong. It can be a protein coat, right? Capsids, capsomeres, etc. So 1 and 2 only are correct. What are features of prokaryote cells, chloroplasts, and mitochondria? So chloroplasts and mitochondria, and also prokaryotes, all of them have ribosomes smaller than ATS. Basically, all these three are prokaryotes. Okay, that's the basic thing. What else? So they also have circular DNA because these are characteristics of prokaryotes. They were bacteria at one point. That's the endosymbiont theory. Now, what about D double membrane though? D. Like it's either between, it can't be A or C. It's either between B or D. So a double membrane structure, right? Basically, you need to understand that mitochondria and chloroplasts, they do have a double membrane, an inner membrane and an outer membrane. Okay, but what about prokaryote cells? Do they have a double membrane structure? Think about it. If we think about the characteristics of a prokaryote cell membrane, they have a plasma membrane or the cell surface membrane, which is a phospholipid bilayer. Okay, but listen, even though it's a phospholipid bilayer, it's still only one membrane. Okay, the surrounding is just one membrane. It may be a bilayer, but it's just one membrane. But if you look at a mitochondria, do you want to know how it's like? It's like this. There's one bilayer outside and there's another bilayer inside. That's the difference basically. So in total there are four layers because there's a double membrane. That's only unique to chloroplasts and mitochondria. Okay, so the answer is B. A student used medic solution to test a sample known to contain carbohydrate. At the end of the test, the solution was blue. Basically, there's no reducing sugar present. It can't be glucose, right? And basically, what's the answer? It's clearly sucrose, right? It's a disaccharide which is not a reducing sugar. But all these three would give a positive test. For 8, sugars with a ring structure can also have a linear structure. Which sugar molecules could be represented by the linear structure in the diagram? Okay, so you guys need to understand something. This is really important. Sugars are of two types, aldose and ketones. Aldose sugars and ketose sugars. So this is an aldehyde, right? Glucose is an aldose, so is galactose. Okay, but fructose, okay, fructose is a ketose sugar. It has a ketone group basically. But glucose and galactose, they are aldose sugars because they have an aldehyde group. So this is glucose for sure, 6 carbon. But it's not ribose or deoxyribose because these are 5 carbon compounds. We have 1, 2, 3, 4, 5, 6 over here. That's why these two are wrong. The answer is D. 8 is D. Which row matches each molecule to a type of bond that is present? The ester bond is present in lipids or phospholipids, right? The hydrogen bond is present in cellulose. So the answer is probably D. And disulfide bond is present in proteins, okay? Antibodies, so it's right. Which shows features of a carbohydrate polymer found inside animal cells. Animal cells, we're looking for glycogen, okay? So it has both 1,4 and 1,6 alpha glycosidic bonds, both of them, and it is branched, okay? This is what we're looking for. 10 is A. Quite straightforward. The statements describe the structure of a polysaccharide found in the cell walls of certain plants. It is composed of 2 different monosaccharides. The monosaccharides are joined by 1,4 glycosidic bonds. The polysaccharide contains pentose sugars. Which statements are true for cellulose? Cellulose is composed of glucose, hexose, so 3 is wrong. 3 can't be the answer. The monosaccharide is joined by 1,4, this is true. It is composed of 2 different monosaccharides, that's wrong. It's only composed of glucose, beta glucose, right? So 11D. For 12, the general formula for a saturated fatty acid is this, CnH2nO2. So let's write down the molecular formulas first. That's going to be better. C11H19 plus 1 is 10 plus 1 is 11, and 19 plus 1 is 20 O2. What about this? C16H32O2, C18H32O2, C19H33O2. Okay. So basically for 2 it's actually double. So this is a saturated fatty acid. 2 is a saturated fatty acid. So the answer needs to be C because they are not double. Okay. What enables triglycerides to perform their functions in living organisms? Triglycerides have hydrophobic and hydrophilic regions. This isn't the function of a triglyceride. That's the function of a phospholipid, right? So 1 isn't correct. Triglycerides have a high ratio of carbon hydrogen bonds to carbon atoms. This is true. That's why we can easily get energy from them. A lot of energy can be stored. You'll learn more about this in A2. Hydrolysis of triglycerides releases metabolic water. That doesn't really make sense, does it now? So the only reason is actually 2. 1 and 3 don't fit in here. And hydrolysis does not release water, by the way. We need water to hydrolyze something. Okay. The diagram shows 3 examples of different bonds. Which bonds can hold the quaternary structure proteins together? In quaternary structure, we have a few bonds: disulfide, hydrogen, hydrophobic interactions, and ionic interactions. So this is a peptide bond. This is for primary structure. We can't get to bond 2. Okay, it has to be 1 and 3. 1 is a hydrogen bond, and 3 is a disulfide bond. Sounds good? 14 is C. Let's go to 15. Which row correctly describes the primary structure, secondary structure, tertiary structure, and quaternary structure of some proteins? Primary structure determines the folding, not really. Defines the order of amino. This is true. This looks good. Involves covalent bonds only. This looks good. Involves peptide bonds between the side chains. These are wrong. Okay. So let's go to the next one. Usually forms immediately after polypeptide synthesis. Okay. Is held together by all the bonds of bonding that occurs in proteins. Okay, so there's one thing I want to say about 14. Quaternary structure, I actually was talking about tertiary structure, but tertiary and quaternary are almost the same. Okay, remember that. So for here in B, quaternary structure is found in globular proteins but never in fibrous. This is wrong. We do find it in collagen, right? So the answer should be C. Check this out. Involves interactions between hydrogen and oxygen, essential for the function of enzymes and receptors. Tertiary, formed when 2 or more polypeptide chains join together. So C is fine. 16. Which relationships could be investigated using a colorimeter? The effect of light intensity on the rate at which a solution of light-sensitive dye changes from green to colorless. This looks good. Anywhere where there's change in color, when there's a change in concentration of a solution, color changes, you can use a colorimeter. Okay, so A is fine. The effect of temperature on the rate of breakdown of cell membranes in tissue with pigmented cells. This is good. The effect of pH on the rate of breakdown of cell membranes in tissues with pigmented. The effect of pH on the rate of release of oxygen from the breakdown of hydrogen peroxide. This is totally colorless. 3 won't do. The effect of light intensity on the rate of change of skin color of lizards that become paler in bright light. So typically we don't use a colorimeter on living objects. Do you guys understand? Like 4 does make sense in a way, but it's not the best answer. We typically use it on solutions. Okay, something that's stationary. So the best one would be for this question B. 16 should be B. That's the best answer. Okay. Okay, which transport mechanism does not require a concentration gradient to be present in order to take place? So basically all of these. Facilitated diffusion is a passive process. It requires concentration gradient. Osmosis requires one. Transpiration also requires a gradient, right? Like we want water to be lost from the leaves, or else water won't be pulled up. Okay, but exocytosis doesn't require one. It's an active process. High concentrations of ethanol disrupt cell membrane structure by denaturing proteins and increasing the separation of adjacent phospholipid molecules. As a result, cell membranes can decrease in thickness by up to 30% and become more permeable. Okay, so when the separation between adjacent phospholipid molecules increases, thickness decreases and it becomes more permeable. Yeast cells release ethanol as a waste product of metabolism in response to increased ethanol concentration in the environment. Yeast cells are able to increase the tolerance of their cell membranes to ethanol, which statement correctly explains a response to ethanol that could account for the increase in tolerance of yeast cell membranes to ethanol. Okay, decreasing the which statement correctly explains a response to ethanol that could account for the increase in tolerance of yeast cell membranes to ethanol. Okay, basically our goal is to reduce the ethanol concentration. Decreasing the ratio of saturated fatty acids to unsaturated fatty acids within cell membranes helps to prevent the tails of phospholipids on one side of the bilayer from sliding past the tails of phospholipids on the other side of the bilayer. Okay. Increasing the proportion of palmitoleic acid, a C16 unsaturated fatty acid, to oleic acid, a C18 unsaturated, in the phospholipid of the bilayer increases the fluidity of the membrane. Activating a cell signaling pathway triggers the unfolded protein response in the endoplasmic reticulum. The unfolded protein response pauses protein synthesis and initiates cell death in yeast cells with a high proportion of misfolded proteins. Increasing the proportion of ergosterol in the cell membrane prevents the accumulation of polar molecules such as ethanol within the cell. Ergosterol in yeast cells has a similar effect on membrane permeability as cholesterol in mammalian cells. Okay, we got a lot of information. Let's try to deduce the answer. Our goal here is to reduce permeability because ethanol increases permeability. We want to reduce permeability. That's our goal. That's a gaining tolerance. So we want to decrease permeability. So let's check this. For D, increasing the proportion of ergosterol. What does cholesterol help to do? Cholesterol basically affects fluidity in this way. At high temperatures, it decreases fluidity, and at low temperatures, it increases fluidity. That's how it actually uh, changes the fluidity. Mainly a few things affect fluidity, okay? The presence of cholesterol stabilizes membranes. The length of fatty acid tails, shorter fatty acid tails are less likely to interact, which makes the membrane more fluid. So the longer the fatty acid chain, the less fluid it becomes. And the presence of unsaturated fatty acids. The more unsaturated fatty acid chains there are, the more fluid our cell becomes. So look at this. If we decrease the ratio of saturated to unsaturated fatty acid chains within the cell membranes, this helps to prevent the tails of phospholipids on one side of the bilayer from sliding past the tails of the phospholipids on the other side of the bilayer. High concentrations of ethanol disrupt cell membrane structure by denaturing proteins and increasing the separation of adjacent phospholipid molecules. This is the key point. This is how ethanol acts. This is the bilayer, right? This is what it looks like. What ethanol does is, suppose there's a protein here. Ethanol will actually denature this protein. As a result, there will be a gap created between the two phospholipids. Okay, and it becomes more permeable. Now our goal here is somehow making them come closer together. So look at A. Decreasing the ratio of saturated fatty acids to unsaturated within cell membranes helps to prevent the tails of phospholipids of one side of the bilayer from sliding past the tails of phospholipids on the other side of the bilayer. So we're talking about side, this side and the exterior side. There are two sides. This response by decreasing the ratio of saturated to unsaturated, saturated to unsaturated. If you decrease this ratio, the number of unsaturated fatty acids increases basically. It does increase fluidity, right? And ultimately, in all of these, we actually increase fluidity, right? At the beginning, I was saying that we need to decrease it to increase our tolerance. But tolerance actually means before ethanol even comes in, right? Or after it comes in, what measures can we take to prevent this denaturation? Yeast cells release ethanol as a waste product of metabolism. In response to increased ethanol concentration in their environment, yeast cells are able to increase their tolerance of the cell membranes to ethanol. Okay? Basically, because they produce ethanol, right? So they need an adaptation to live in that environment. Okay? So they somehow need to fix this problem. See, this as a result, cell membranes can decrease in thickness by up to 30%. Thickness decreases and becomes more permeable. This was the previous bilayer. Now it becomes like this. It becomes much less thick. But doing this, why A is the answer, I'll exactly tell you. If we decrease the ratio of this, it helps to prevent the tails of phospholipids of one side of the bilayer from sliding past the tails of phospholipids on the other side. And as a result, this decrease in thickness won't occur. Okay, so in the context of this question, A is the best answer. That's the logic behind this. These are fine. Okay, like if you do increase the proportion of C16, a shorter fatty acid chain, right? Both of them are unsaturated. Fluidity does increase, but it's just not the best answer. Okay, let's go to 19. In an investigation, a plant cell was placed in pure water. The initial rate at which water molecules entered the cell R was greater than the initial rate at which water left the cell. Okay, so water was entering the cell. This was greater than the rate at which water left. In a second investigation, a plant cell of the same type was placed in solution with a water potential equal to that. What will happen in the second investigation over a period of 5 minutes? This is wrong. Water enters and leaves, but at the same rate. Okay, water molecules will enter and leave the cell. A is wrong because it's saying that water does not enter. So in BCD, these are all right. Will enter and leave the cell in equal amounts, both at an initial rate that is less than R, both at an initial rate that is greater than R, both at an initial rate that is equal to R. So if you place it in pure water, basically the water potential outside is much greater. That's why water will enter the cell at a very fast rate. Do you understand? But when you put it in a solution that is equimolar, that has the same concentration as this, the rate of movement won't be that fast. It will be much slower. Okay, that's why it should be less than R, because the concentration gradient is less steep. All right. The protein P53 is produced in a cell in response to DNA damage. This protein stops the cell cycle for a short time just before the DNA is replicated. Okay, so DNA is replicated in the S phase. This occurs before replication. So it should be G1, just before the S phase, right? 21. Some parts of a typical human chromosome are more numerous than others. Which parts are listed in order from the most numerous to least numerous? Okay, most to least. We have a lot of histones. We have a lot of histones. Centromeres are not that numerous, okay? It's not A. It should be at the last, okay? Centromere should be almost at the last comparatively. DNA molecule, telomeres, centromeres. No, DNA molecules aren't the most numerous. We don't have that many, okay? Histones, there are a lot of histone proteins because for each chromosome, right, we need multiple histone proteins. It's very numerous, right? Followed by telomeres. They're at the end of each DNA molecules, right? Two for each, and then we have the DNA molecules. This makes sense. C is the best answer, right? And D is wrong. D is wrong. Where is the centromere located, by the way? It's at the center, right? There's one centromere. Here we have one centromere. This is the one centromere. We have two DNA molecules, right? And for each DNA molecule, we have two telomeres at the end. That's the logic, basically. That's why C is the answer. One DNA molecule, two telomeres for each DNA molecule, and multiple histone proteins to wrap it up. Which row correctly describes the stage of mitosis? Prophase, nuclear envelope disappears. That's fine. Centromeres replicate. Spindle microtubules begin to form. The centromeres have already replicated before prophase. Okay, this is not right. Metaphase, nuclear envelope is not present. True. Moves to the poles of the cell. Not really. This has occurred before metaphase has started. Anaphase, nuclear envelope begins to reform. No, it begins to reform in telophase. Okay, begins to reform and reforms in telophase, not anaphase. Okay, in telophase at maximum distance from cell equator. True. Spindle microtubules breakdown. D looks good. 22 is D. So which ones were wrong here? Basically, these have already replicated. These have already moved to the opposite sides. There's a brief period between prophase and metaphase. Some things happen then, right? And yeah, this is wrong. The diagram represents a nucleotide containing adenine. Which statement about this nucleotide is correct? Adenine is a double ring structure, so it's a purine. The carbohydrate is a pentose sugar. This is true. The base contains nitrogen, of course. Base pairing occurs with uracil. Adenine does bind with uracil, but not in DNA. In RNA, true. Adenine is a pyrimidine. No, it's a purine. So 1, 2, and 3. 23 is B, okay? Oh, by the way, guys, about 18, like C and D don't make sense, right? C and D aren't even in the question. About B, since we've changed, since we've changed um, a proportion, I read this wrong actually, a proportion of C16, we've changed C16 to C18. If you increase carbon number, fluidity actually decreases. That's why this statement was wrong. Fluidity does not increase, fluidity actually decreases. And about C and D, C doesn't make any sense because this won't increase tolerance of one cell. And D, increasing the proportion of ergosterol in the cell membrane prevents the accumulation of polar molecules such as ethanol within the cell. This has no connection really. Okay, so A is the best answer. B would have been fine, but B is a wrong statement. Okay, so A is the best answer. Moving on to 24. Which row is correct for the replication of a DNA molecule? Um, so DNA polymerase works in this direction. Remember, it always goes from the 5' to 3' end, right? And the enzyme is DNA polymerase. So the answer should be C. What does ligase do? It seals the nicks, right, between the Okazaki fragments. For 25, some students were asked to look at the photomicrograph of a cross-section of unfamiliar material and describe what they could see. So this is basically a cross-section. Do you guys understand? So we know for a fact that it is circular because it was like this and we cut it like this and we're looking at it from above now. So we can say that it is circular. But we don't know about the 3D shape, okay? We can't say that it's spherical, but we do know that it's circular. Now we don't know if it's hollow. There might be content inside. We don't know from this photomicrograph. Okay, it might be hollow if it was xylem or something, like if it was dead, but it still may contain liquid, right? So we just know for a fact that it is circular. So the answer should be C, circular only. The diagram shows the relationship between phloem safety elements, xylem vessels, and companion cells. Which row identifies what could be represented by 1, 2, 3, 4, and 5? So companion cells have endoplasmic reticulum. Phloem sieve tube cells also have endoplasmic reticulum, but they don't have a nucleus, right? And they have scanty cytoplasm. Xylem vessels do not have a nucleus as well. So A is correct. I'm going to tell you why the others are wrong. Xylem does not have cytoplasm. Xylem does not have nucleus. Phloem sieve tube elements do not have. Wait, so xylem does not have cytoplasm, but phloem sieve tube does have cytoplasm. Remember that. It may be scanty, but there's cytoplasm, and it doesn't typically have a vacuole. Okay, so the best answer is A. 27. Why does an air bubble in a xylem stop the flow of water? Basically, what the air bubble does is, look at this. This is xylem. This was the flow of water over here. This is the flow of water. It's supposed to go up. Now there's an air bubble here. What does it prevent? Just look at the issue. Water can still stick to the walls. Okay, so there's no loss in adhesion. This is wrong, but there's a break in the contact between water above and below. Do you understand? That's why there's a loss of cohesion. So 2 is the one. Air bubbles do not cause collapse. The answer should be 2. Okay, but there's no loss of adhesion, by the way. Which changes to the water potential and the volume of solution of phloem tube occur when amino acids are moved into a sink from the phloem sieve tube. Okay, so this is the phloem sieve tube. Now it will move into the sink or the roots or something. Now amino acids are moved into a sink. Amino acids move here. After the amino acids move, what happens to the volume of solution in the phloem sieve tube? Think about it. Since water potential of this will increase, water will follow the amino acids, right? And water inside the phloem sieve tube will actually decrease. But since we lost amino acids from the phloem sieve tube, right? We'll just lost solids. What will happen? The water potential will become higher. Okay, so you should think of it this way. This is what happens. Basically, we have the phloem sieve tube. This is our root, maybe. So as we lose amino acids, solids decreases, so water potential will actually increase. We just lost some solids. But since water potential is higher, lower here and higher here, water will follow and go from left to right, and volume decreases. Okay, do you get it? For 29, which feature of some xerophytic leaves reduces the rate of transpiration by decreasing the water potential gradient between the internal leaf surface and atmosphere? So 29A isn't correct. That there's no change in the gradient due to the waxy layer. Leaves reduced to spines with a small surface area to volume ratio. This has no effect on the gradient. It just reduces the surface area to volume ratio. So less diffusion occurs. Stomata located in sunken pits. This is good. Look at this. If the leaf is like this and the stomata are like this, right? What's going to happen? It's going to create a region of high water potential here. Water potential will also be high here, so the gradient will be reduced. That's what happens in this xerophyte, right? 30. The diagram shows a cross section through a mammalian heart. Clearly, you can see that the wall is thicker over here compared to here. So H is clearly the ventricular side. Okay, H is clearly the ventricular side. That is why B is wrong. It's the left side, I mean, okay? H is the left side. My bad. H is the left side. So wherever, like, if you look at the heart like this, it's divided into 4 sections, right? Like this. The atria are above and the ventricles are below. But the walls of the left side are thicker because it has to supply blood to the whole body, right? So for H, it can't be the right side, it needs to be the left side. So we're getting rid of A and C. Now, are we seeing the cross section of the atria or the ventricles? This is the most important part. That's I was why did I cross out B? Because this is clearly not the atrium. This is clearly the ventricle. Okay. How am I so sure? Check this. Wait. Basically, if you looked at the atria, you would see the valves. Wait, I'm getting a good figure for you guys. This is what it would look like, something like this, basically. We would see the valves, okay? But here we aren't seeing the valves. If you ever look at this picture, if you ever see this picture, remember that this is of the ventricles, and it's just that they've shifted the left and right side to trick you guys, okay? This is actually the left side, but you know, when we look at the heart, the left side is typically on the right. You know, it's laterally inverted, right? So for 31, blood entering the heart from the vena cava passes through or past several structures before entering the lungs. There was a similar question in paper 2. After arranging these 5 structures in the correct order of blood flow from vena cava to lungs, which structure will be third? Okay. So let's 5 of these structures are included in the list. From vena cava before entering the lungs. So from the superior and inferior vena cava, we are first going to be at the right atrium. Okay, we're going to be at the right atrium, not the left atrium. Okay, then we're going to pass through the tricuspid valve. Is it here? No tricuspid valve. Then we're going to enter the right ventricle. After that, we're going to enter the pulmonary artery. We're going to enter the pulmonary artery. But before entering the pulmonary artery, we need to pass the semilunar valve. Do you guys understand? Now, here's the thing. Some structures are totally going to be excluded. Like, we don't need left atria here. We don't need aorta here. Okay, we don't need this because that's on the left side of the heart. And we don't need pulmonary vein because we'll be crossing the lungs that way. Okay. But what's important here is that we have some tissue, conductive tissue of the heart, right? That actually produce the impulses. So what's the first thing that you're gonna pass as the blood passes through the superior vena cava? Basically, in the right atrium, we know that we have the SA and the sinoatrial node. This is number 1. Okay, this is number 1. What's next? As we pass through the tricuspid valve, just over here in the septal wall, you know there's another node, that's the atrioventricular node, right? Followed by that, in the ventricular septum, what do we have? We have the Purkinje tissue. So this is number 3. Followed by the pulmonary artery, followed by the semilunar valve, I mean, and then the pulmonary artery. This was the pattern. So at 3, we have Purkinje tissue. So the answer is B. 31 is B. The bar chart shows the quantity of endothelial tissue, elastic tissue, and muscle tissue in the walls of 3 vessels. Okay. So remember, we have the highest elastic tissue in the arteries that are close to the heart, basically aorta. So the more elastic tissue we have, this one, elastic tissue, we are an elastic artery. So P is clearly inelastic artery. So it's between A and D. Now for muscle tissue, for muscular arteries, muscle tissue will be the highest. This is muscular tissue. Okay. So what's the difference between Q and R? Both have high muscle tissue, but Q has higher elastic tissue as well. So, and endothelial tissue is just a single layer. So Q is the better fit, right? Q is the better fit, and R doesn't have such a thick, tunica intima, media, and externa. So 32A is the best answer. For 33, the maximum pressure in each of the four chambers of a healthy human heart was recorded during one cardiac cycle. The maximum pressures recorded were 3 millimeters of mercury, 10, 25, and 120. Which value was recorded for the right ventricle? So listen, where do we have maximum pressure? In the left ventricle, right? The right ventricle, it's not as high as the left ventricle. Remember, the left ventricle is at 120, but it is still high. This is the left ventricle, right? So it's the highest. It's not the highest, but it's very high comparatively compared to the other two. So it has to be 25. See, if it was left ventricle, it would be 120. As we go further from the heart, pressure slowly decreases. Okay. As you can see here, the graph shows the pressure changes in three structures of the right side of the heart during the cardiac cycle. What is represented by line T? This is really important. This is really important. So 434, we first need to know what this dotted line is, okay? The highest change in pressure. I just taught you guys. What is this? From this section. Like, this is the these are for four chambers, right? But this is for the right side of the heart. Now, you see this structure, the bold line, the line labeled T. Remember that this is the pulmonary artery. Okay, this is the pulmonary artery. I'm going to show you a figure. Wait, this is really important for you guys. You need to memorize each and every one. Here you go. Wait. Okay. So basically, the dotted line that represents the ventricular pressure, okay? The one I marked with an arrow. This is ventricular pressure. And this one over here, the bold line T, it represents the aortic pressure, or you could say that it's the pressure in the pulmonary artery, okay? Now, do you see the line below? There's another dotted line that represents the atrial pressure, okay? So these three lines are very, very important, okay? So line label T is the pulmonary artery. Next, which statement helps to explain why there is no cartilage in the walls of bronchioles? Why do we need cartilage in the trachea? To keep it open. So in bronchioles, we mainly have smooth muscles. They contract and relax to, regulate the flow of air. So D is wrong because the smooth muscles change shape. Gases must diffuse. This is also wrong. C is the best answer. A, cartilage would make the bronchioles too narrow. This doesn't make sense. It would actually make them too wide. Okay. For 36, the photomicrograph shows the cross section of part of the gaseous exchange system of a mammal. So, do you know what this is? This is one alveoli over here on that side. This is an alveoli. These are all alveoli. So why is V is not an alveoli clearly? So if you look at the respiratory system, we have the trachea, bronchus, bronchioles, then we have respiratory bronchioles followed by these. So clearly this is not a trachea, this is not the trachea, this is not the alveolus, it's clearly the bronchiole because we get alveoli from bronchioles, okay? 37. Which feature of the disease cholera decreases the spread of the pathogen Vibrio cholerae? Immunity to cholera after vaccination is short-lived. So this doesn't help really, right? 37 is not a. Up to 98% of people infected with Vibrio cholerae symptoms. This is wrong because it helps in the spread. We don't know who are cases or carriers, okay? Cholera rapidly kills up to 50% of people with symptoms if they are not treated. This is correct. Like if the person dies, they can't transmit it, right? That's the thing. Therapy is successful and treats out 99% of people with symptoms of cholera. So for D, it's good for treatment, but it doesn't control spread. Okay, to control spread, basically it's the fecal-oral route, right? We don't want treatment. We want some way in which the transmission cycle is broken. So C is the better answer. All right. For 38, which row shows how penicillin kills bacteria? Mainly, it prevents the formation. It prevents the formation of the crosslinks, right? Remember that to grow, the bacteria itself breaks down the cell wall. But then it wants to form those again after taking in water and expanding, right? But remember that after making the holes, bacteria using autolysins, they want to create the bonds again. But penicillin actually prevents formation of these bonds, right? So it actually prevents the formation of the peptidoglycan crosslinks. So it's between A and C. So water will enter the bacteria and burst. In the hybridoma method, what is grown by cell culture to produce monoclonal antibodies? So mainly we combine the cancer cells. We combine the cancer cells so that we get immortal cells, right? So what's the method like exactly? We first inject the antigen into the rat or sample, the animal. So the rat actually produces antibodies. Then we combine the B lymphocytes and myeloma cells to get the hybridoma cells, right? So we actually combine the myeloma cells, the cancer cells, with lymphocytes. Do you understand? So lymphocytes plus the myeloma or cancer cells gives us the hybridoma cells. So in the hybridoma method, what is grown by cell culture to produce monoclonal antibodies? So it's not the myeloma cells, okay, because we are combining them with lymphocytes. It's not these two, and we inject the antibody into the organism, so it has to be clones. Hybridoma cells are the clones basically. What type of immunity does a baby have at birth? From the mother, it actually gains natural passive immunity, okay, through the placenta. Natural, passive, passive, natural. So that's it. So yeah, if you have other questions, you can ask me, and I'm gonna complete the 2021 series fully, all 7 variants, very soon. I'm gonna link the paper 1 playlist over here, right? And I'm gonna link the video for Feb March 2022 paper 2 over here, okay? And remember to subscribe to the channel, and if you have other queries or requests for papers, you can just let me know. I'll do them in my free time, okay?